All right, well, welcome everybody. Today's speaker is Dr. Ingrid Spies. She's currently a research fisheries biologist at the Alaska Fisheries Science Center here in Seattle. And Ingrid has considerable expertise in genetics. She's worked on a number of species. Today, she's going to talk about probably 2 or 3 species. But a lot of her work, I think, has been related to cod genetics. She has a PhD from the University of Washington, the School of Aquatic and Fishery Sciences, like a lot of people here. And she's also done work internationally, more recently at the University of Oslo in Norway. So it's a pleasure to have you here, Ingrid. We look forward to the talk.
Thank you. Thank you. Thank you. Does that sound okay? Sounds weird to me. So yeah, today I'll be talking to you about some recent work I've done using next generation sequence data, and I'm excited to see where you all go with regard to the future of genetics and halibut. I was putting together this talk and I realized that the 3 kind of topics of the different species I was working on were kind of funny when you put them all together. Pollock, cod, and skates. So pollock, cod, and skates are 3 species that are really commercially important where I work at the Alaska Fisheries Science Center. Pollock is the largest fishery in Alaska, and Pacific cod, I believe, is 4th, and I think it might be 4th after pollock, crab, and I think halibut. And then finally, I'll be talking about some Pacific skates, which may not be as important as a fishery, but they are very important to understand because they are long-lived, late to mature, and low fecundity. Now, I think that I'm going to start this talk with a little bit of a timeline about why I think this is a really exciting time for the field of genetics and applying it to fishery science. This whole graph, actually, I realized today spans my lifetime. So in my lifetime, so much has happened. The first person you see there is Fred Utter, who many people consider to be the grandfather of fishery science. And he first applied allozymes, or protein electrophoretic markers, to fish in the 1970s. Later in the 70s, Sanger sequencing was invented, but I'm kind of struggling to understand how you could have Sanger sequencing without PCR, but that's what I saw when I did my Google search. In the 1984, Carrie Muller invented PCR, which was an amazing development. It allowed people to amplify many, many millions of pieces of a certain part of the genome they were interested in. That was an incredible tool. I think he might be a billionaire by now. And then with that advent of PCR, microsatellites became very useful. And so when I started working on fisheries science in the 90s with Paul Benson, he was using microsatellites as a tool. And microsatellite is also called a short tandem repeat. Like if you imagine a little bit of the genome that's a repeat of like GATA, it would be a repeat of, say, 4 might be predominant in one population and a repeat of 6 might be predominant in another one, and that could tell them apart in some sense. Later on in the 2000s, SNPs became more prevalent. And I have a picture of Lisa Sieb here, who is a researcher who has done quite a bit of work applying SNPs to various species of salmon. Here's what a SNP is, essentially, if you're not familiar with it. You know, DNA is coded with 4 base pairs, G, A, T, and C. And here you see 2 sets of DNA, maybe they're 2 individuals, and you can see the base pairs, you know, A codes with A base bonds with T and G bonds with C. And they're pretty similar between the 2 of them, but here you see this one site, the C and the G are different from the other individual, which has a T and an A. That's a SNP, a single nucleotide polymorphism. And it doesn't look like much, but once you gather thousands of those and you compare, you know, whatever you're interested in, it can add up to something that can tell you a lot about different aspects of a species. So SNPs became popular, but not long after that, this guy, I believe it's Nick Cook, invented something called next generation sequencing. I think he could have come up with a better name because now there's next generation everything. And I bet he did it in a team because this is a big deal. But essentially, this allows cheap, fast DNA sequencing, actually RNA as well. Let's put this in perspective. The Human Genome Project, before next generation sequencing was invented, took 15 years and cost $2.7 billion. Today, I could sequence one fish in less than 2 months for $1,000. It's so cool. So this is why I think we are at a very, very exciting time for this field. It's accessible. You can get more data than you know what to do with. and the price is really reasonable. And so there's been quite a bit of work in all these types of genetics that I talked about on Pacific halibut. There was a study even with allozymes that Stu Grant wrote back in, I think, 1984. There's been studies using microsatellites, probably not a population structure study using SNP data, but quite a bit of work has been done with halibut. And I think that the future holds also a lot of potential for halibut. The one thing about genetics is what they say is absence of evidence is not evidence for absence, meaning that if you don't find something using genetics, you might not have had enough statistical power to find it. But on the other hand, if you're using the whole genome and you don't find anything, it's probably not there, which is another reason this is a really exciting time in fisheries genetics. So several of the projects I'll be talking about today use something called RAD sequencing, restriction site associated DNA sequencing. This isn't whole genome sequencing. Instead, it's sequencing a range of locations throughout the genome, kind of randomly spread out throughout the genome, but they're not random in the sense that you can compare your results among individuals because it uses one enzyme called a restriction enzyme to cut the genome up into these little bitty pieces where it sees this particular pattern. And this pattern, you think, well, that's pretty specific, but the genome's also quite large. So you might get as many as 20,000 or more, actually, sequences from RAD sequencing. So it cuts up the genome, and right where all these cut sites are, you get a short bit of sequence, maybe 100 to 150 base pairs. And then you take your data, and the nature of next generation sequencing is you get quite a few sequences, and you can line them up. These are called stacks. And if you get a stack of a certain depth, you can say, well, that's probably real because I got so many sequences and they line right up. And so it's not just some artifact. And so that's the method you use to kind of ground truth your data. And what's cool is since these cut sites are shared among individuals, you can compare the RAD sequencing sites among individuals, even among species. So my first topic is, I called it Wandering Cod, but here's the official title. Genetic evidence of a northward range expansion in the Eastern Bering Sea stock of Pacific cod. And this is a project that I worked on with quite a few people. And I think it's an example of how fisheries genetics can be really effectively used to help fisheries management. So this all problem started when we started looking at the 2017 results of the 2017 Northern Bering Sea Survey that the National Marine Fisheries Service put on. The last Northern Bering Sea Survey they had done was in 2010. And we noticed a 900% increase in the abundance of Pacific cod in this region here. I need to orient you all. This is Alaska, Aleutian Islands, and this is the Bering Sea. We noticed so many more Pacific cod in this region of the Northern Bering Sea than we had before. This is the Eastern Bering Sea shelf, where we usually see Pacific cod. The Eastern Bering Sea shelf since 1982 has been the only survey that has been fed into our Eastern Bering Sea cod stock assessment. And so there were so many cod here, we were like, where did they come from? This is so strange. And at the same time, the abundance in the Eastern Bering Sea declined by 37%, which was the largest decline in the survey history. And as I was sitting there hearing this, I said to myself, oh my goodness, I took a genetic sample on that survey of cod. I was so excited. So here's the 2018 data thrown into this. So you can see 2010, there's almost no cod. So yeah, the star is the Northern Bering Sea Survey. 2017, there's quite a few. And 2018, there's so many cod in the Northern Bering Sea that there were more than we saw in the Eastern Bering Sea. And this time series is the Eastern Bering Sea cod survey estimate. Now, at the same time, we saw a very large decline in Gulf of Alaska Pacific cod. The lowest estimate on that 2017 survey was the lowest in history. And Steve Barbeau, the stock assessment author, hypothesized that this was due to warmer temperatures. When fish are in warmer water, they need to eat more. And so his idea was that their metabolic demands were higher than the amount of food that was available. So there's high natural mortality. So putting all this together, the question came up, where did all these cod in the Northern Bering Sea come from? Did they come from the Gulf of Alaska cod? Maybe they moved north. Were they from Russia? Russia's right up there. Were they a population that was already there that maybe grew? And so I knew that there had been a recent study by Dan Drynan on population structure of Pacific cod. And so I was able to use baseline data from his study. He had Prince William Sound, Kodiak Island, and ADAC rad sequencing baselines. Essentially, enough SNPs that we kind of had a genetic baseline, what we knew these spawning populations were like. And then I threw in a Unimac, Pribilof, and Pervenets sample. And these were our baselines of spawning fish that we used to compare this 2017 sample from the Northern Bering Sea to. And I need to tell you a little bit about the life history of cod because it's really, really important here. We believe they home to spawn in the winter between February, March, and April. You'll see them going to their spawning grounds, spawning, and then dispersing. Who knows where? We don't actually know where. About half the fishery takes place on the spawning grounds and half the fishery takes place in the summer. So this is a little bit of a management conundrum because we do our survey in the summer. But that's important here because these were taken in winter and these were taken in summer. And here's a map that Sandy Neidicher put together on observed spawning areas of cod by observers throughout their range. So you can see they spawn up in the Eastern Bering Sea, in the Aleutian Islands. They definitely spawn in the Gulf. We've never, that I know of spawning up here. So I did a rad sequencing study. I'm not going to tell you a whole lot about the details, but I'll tell you the results. There was about 3,500 SNP loci in this study, and I did something called an assignment test. And I took these Northern Bering Sea fish, and I said, okay, of these reference samples that I showed you, the spawning groups, what appears to be the most likely group that they're most similar to? And in the Eastern Bering Sea, remember, there was 3 samples, Unimac, Pribilof, and Pervenets. So the first test I did, I kept all the samples. Here's all the spawning populations. I kept all of these in as reference populations. And the output of the assignment testing program said, okay, I think there's 21% of them are from Pribilof, 31 are from Pervenets, and 46% are from Unimac. And all the other ones had a posterior probability of almost 0. So that answered a lot of the questions. It seemed like these Northern Bering Sea fish were actually Eastern Bering Sea fish that had moved north, further north than ever before. And then I also said, okay, well, what if we just combine the Eastern Bering Sea spawning reference samples into one? And when I did that, it gave me a 100% probability that the Northern Bering Sea fish came from Eastern Bering Sea spawning stocks. I think I'll skip this slide. This is another way of looking at the data. This is called a discriminant analysis of principal components that takes the results and shows them in a spatial manner based on the eigenvalues associated with the different, all the SNP loci. And so individuals that are more closely related will group together. And what you see, what really pulls out here, is that there are some spawning samples that really seem distinct. Adak seems quite distinct, Prince William Sound very distinct, Kodiak very distinct. And then you see this lump of rainbow here. But what this is are all the Eastern Bering Sea samples grouping with the Northern Bering Sea. And just to see if there was more resolution there, I took out the samples on the left, kind of zoomed in here. And you can see that the Northern Bering Sea sample clusters really closely with Unimac Pribilof, and you can't see it here, but Pervenets. So that, one more thing. This is a plot of a lot of statistics, and I'm not going to go over a lot of them, but here's all of our sampling locations. And one of the statistics I looked at is called allelic richness, which is essentially the number of alleles in each sample. And remember earlier I said, could this be a founder effect where perhaps there was a very, very small population that rapidly increased, maybe conditions were really favorable? And if that happens, oftentimes people assume that a signature of a founder effect is when you have kind of low allelic richness, low genetic diversity, because it started from just a few fish. And if you look at the Northern Bering Sea sample, is 1.97, and all these are very small, very close together, but it's actually higher than the allelic richness from Pervenets, Pribilof, or Unimac, which makes me think more that it's a mixture of those different groups than a founder effect. So I ruled out the founder effect. Actually, the other thing I think I ruled out is, well, the other consideration that a lot of people wanted to know is, could these fish be from Russia? And I looked at this quite a lot of different ways, and I think that the highest probability is no, they just came from Eastern Bering Sea spawning groups in our waters. But there also are Russian spawning fish, I think, along the Eastern Bering Sea, just outside the international line, that I would imagine are pretty similar. I mean, the spawning groups along the Eastern Bering Sea shelf, Pervenets, Pribilof, and Unimac were quite similar, so I would not be surprised if the Russian fish are quite similar too. So my answer to that is I can't rule it out, but I think it's unlikely. So what was great about this study is that I was able to give the results to the stock assessment author in May or June, and the stock assessments are due in September, October, November. And so he was able to incorporate this information and use the Northern Bering Sea Survey in his assessment for the first time in the whole time series. And now I want to show you a little bit more about what we know about cod tracking temperatures, because this whole story is related quite a bit to temperatures. Here's the bottom temperature of the Bering Sea in 2010. In this bluish and hot pink area is something known as the cold pool, which is left behind after the ice retreats in the Bering Sea, and cod avoid it. And you can see here from our 2010 survey that the cod distribution is highest outside of it. Actually, there's almost no cod inside of this cold pool. And you see the same thing in 2017, but in 2017, there was less of a cold pool. And there's this kind of corridor of warmer water. And sure enough, looks like the cod might have tracked up through this warmer water here, maybe for feeding. We're not sure why, but I've heard they're kind of fat and happy up there. And And then again, so there was a cold pool in 2016, 2017, but the big news in 2018 was that it was the first year of our survey that we saw no cold pool, which might explain the movement of cod up north as well. And we know that the Bering Sea is changing. There's been many, many studies that have shown this. Jackie Griebmeier wrote a paper in Science back in 2006 that documented that the Bering Sea is shifting from, actually I say by now it has shifted from an Arctic to a subarctic ocean, between the less seasonal sea ice, higher ocean and air temperatures, and fewer marine mammal and the benthic prey that the marine mammals feed on, and they've been replaced by pelagic fish. So this story is quite linked to the ecosystem and the climate. And studies on climate change do predict that we will see higher shifts in northern shifts in all species really, but here I believe it's been documented in cod. So I believe that there's some really interesting implications of this. If cod regularly spawn in the Eastern Bering Sea, and now they're shifted 1,000 to 2,000 kilometers further north, what would this do to spawning? Are they going to swim all the way back down? Or will they find another spawning site that's closer? We don't really know, but what we do know is reproduction is really low in cod right now. For the past 4 years, we've seen lower than average reproduction, and we're really interested to see what's going to happen in 2019. But something has definitely changed here. And their overall, their body condition also seems to be low. This is on the y-axis, this is a length weight residual that's used as a measure of fish condition. And we don't have a lot of data in the Northern Bering Sea on fish condition, but I actually think that they're doing great up there, just based on what the fishermen tell us. And yeah, fishing has increased up there. Here's plots of cod hulls and their location in the Eastern Bering Sea. And you can kind of see it creeping north, particularly in 2018. This is St. Lawrence Island. And I've heard reports of lots and lots of cod kind of aggregating south of St. Lawrence Island. So I'll sum up this kind of part of the talk here, that looks like the increase of Pacific cod in the Northern Bering Sea is just movement of Eastern Bering Sea up north, likely due to shifts in ocean temperature. And they're likely not from Russia, the Gulf of Alaska, or anywhere else. And I'd be happy to take any questions throughout the talk. But otherwise, I'll keep going to the next piece of research I want to tell you all about. So we're moving on from this study, which was rad sequencing, to whole genome sequencing now. And we've done whole genome sequencing on the samples you see represented here. There was a temporal replicate of 2 of the samples from Unimak and Kodiak Island. And this data has actually been aligned now to the Atlantic cod genome by Carolyn Tarpey, who's been fabulous. She's working with me on this project. And I'll show you a little bit of the preliminary results. This is the number of SNPs that align to different linkage groups of the Atlantic cod genome. And for those of you who are not familiar, what's kind of going on now is people are sequencing the whole genome of like a representative of different species, and they're annotated, meaning as many genes as possible are annotated with their gene function, what they are. And so it's super useful. Say you have a species that's related, you can say, oh my goodness, there's something really interesting going on at this gene right here. And you can actually know which linkage group it's on. And linkage groups are similar to which chromosome. Here, actually, linkage group 24 is the mitochondrial genome. And so it's really neat, all the things you can do with this. I believe Dan Dryned mapped reproduction in halibut to linkage groups to learn more about how sex is determined in halibut. And here is for each of the linkage groups, for the Pacific cod whole genome data, how many SNPs aligned along the linkage group. And what's kind of interesting are places where there's actually nothing in Pacific cod. That's kind of weird. Maybe there's like an inverted part of the chromosome. We have a lot to learn. And what we're going to do with this whole genome data is develop another suite of SNPs that are extremely informative. Like I said, when you have the whole genome, you're not probably going to miss anything. So we're coming up with maybe 500 individual SNP markers that we can use to classify the different spawning areas throughout the range of Pacific cod. And then remember I said we don't know how they're moving? Well, we have a lot of summer samples. A lot of you actually have probably helped collect them. Thank you very much. And along with otoliths in many cases, so that we can take a summer cod and say where it probably was spawned. We can say its age, so we can start to learn about where they're migrating and at what age they're migrating, and really start to hone in on what we really don't understand about cod migration. So this is really exciting. I think we'll have it's called a GTC panel. We'll have our panel of these 500 SNP loci hopefully by this fall and can start using them. Okay, so moving on to a new topic. This one's called hot pollock, but the official title is heterosis as a mechanism for adaptation to high temperatures in a single generation walleye pollock. And this is a completely different study, but it uses the same technology. Heterosis, for those of you that are not familiar with it, is increased fitness in crossbred individuals. It's kind of similar to hybrid vigor, if you've ever heard about that. And in this case, the quality that I'm using for heterosis is heterozygosity. So you inherit one allele from each parent. Well, say you inherit a big A from mom and a big A from dad, that's homozygosity, that's the same allele from each parent. But if you inherit little a from mom and big A from dad, that's a heterozygote. So we designed a study a while back to look at what will be the effect of increasing temperatures on walleye pollock. Obviously, this is a very important, hi Ian, fish in Alaska. And maybe we could learn something about how they will respond to climate change in an experimental setup. So we took one female and 3 males from Shelikof Strait off Kodiak Island, and one female and 3 males from Puget Sound, Washington. Took the fertilized eggs in a thermos, brought them to the Alaska Fisheries Science Center, and reared them at 3 different temperatures that represented temperature extremes to this species. High of about 12 degrees, a medium temperature about 5.8, which is actually a little higher than the ambient temperature at Shelikof Strait, and a low temperature of 1.5 degrees. And then we took the samples out at different time points. There's actually higher mortality at the higher temperature. And these are the time points. The days since spawning are on the x-axis. So we had a bunch of larvae getting reared in these different temperatures. They hatched out from their eggs, and then we sequenced them. There was about 100 from Puget Sound and 100 from the Shelikof Strait area. And since they were a common garden experiment, we didn't really know how to make equal numbers from each family. So we put equal number of eggs in each family, and then when they were sequenced, we could assign them to parentage. And so at the top, you can see the Shelikof Strait families reared at low, medium, and high temperatures, and the different shaded colors represent which male sired which offspring. And you can see it's not really even. Like male 8, there was a lot. Male 9, well, it was only the medium and high temperatures. Male 7 had a low and a high temperature. Same with Puget Sound. We did what we could. And this was an interesting data set. I spent a lot of time looking at it because the research question was, is there a difference at increasing temperatures in these offspring? And I really had no idea what to look at. I looked at this data set for so long, but I did come up with something. So we had approximately 20,000 SNP loci after we filtered. We filtered to a stack depth of 5, which is about the minimum respectable stack depth you would like. But I wanted quite a few loci. We removed individuals that were genotyped less than 70%. Overall, we had about a 95% genotyping rate. And we made sure that we only kept loci that were scored in 75% of populations and 75% of individuals. And here's that result I was showing to you. This shows along the y-axis a measure called FIS, which is essentially a measure of heterozygosity that I was talking about earlier. And along the x-axis, we have temperature. So you see that the families reared at low, medium, high temperature. And you can see a general trend of FIS decreasing that's statistically significant. When FIS decreases, heterozygosity increases. So what this showed was we saw a significant increase in heterozygosity with temperature, which is a really interesting result because this means that when they're stressed in a single generation, pollock are going to adapt, and they're adapting by essentially holding on to their genetic diversity. These fish that have higher heterozygosity, that kept the genetic diversity from both parents, are the ones that survive. And the fact that it's in a single generation gives me a lot of hope for this species. I think they're going to be okay under climate change, at least in the temperatures that we've subjected them to so far. Then I did something where I kind of took advantage of the fact that we had an annotated genome. What I'm showing you here are how the loci associated with this shift in FIS, or increase in heterozygosity, how they align to the linkage group. I'm going to show you first all the loci we used in the whole study, 22,000, and which linkage groups they assign to. So you see kind of a spread between the 23 linkage groups, most on linkage group 1, a lot on 10. Now let's take a look at the Alaska fish. There were 679 loci that increased in heterozygosity with temperature. And what you see is kind of a spread across all of the loci, with a lot on linkage group 1. And in Puget Sound, you also see a spread, but maybe a lot on linkage group 10. So in general, I think this means that this effect is spread throughout the genome, but there might be something going on in particular with the Alaska linkage group 1. There's a gene called pantophysin that is known to be related to temperature in gattids, and it is on linkage group 1. So I'm thinking that that might be something in the future to look at. And then I went even further and I took the genes, I kind of painstakingly went through the data and I found all the SNP loci associated with temperature that were annotated. Remember I said you could get, genes that were interesting. And then I looked at all of them to see which one might be associated with thermal regulation. So what you have here is a set of 4 categories. SNP loci, stop me if this is too complicated, because it is a little bit complicated. So these are all the SNP loci from Shelikof Strait, Puget Sound, and then the third category is from everywhere that showed this increase in heterozygosity with temperature. And then the fourth category, just the white, is throughout the entire genome data set. So this one had just all the loci, nothing interesting about them. So I wanted to compare, was there any genes at which it seemed like it might be related to thermal regulation in pollock. And the one that looked interesting here was disintegrin. You can see that in Shelikof Strait, Puget Sound, and across all families, it was pretty highly represented as showing a shift in heterozygosity. But in the whole data set, it wasn't highly represented. So anyway, that might be a point of future research to look at this as a protein, a gene potential for thermal regulation in pollock. So next I'm going to talk about a category I called faithful skates. And this really is a horrible joke because they're only faithful because they're returning to their spawning site of origin. So this is a study that I worked on recently with quite a few folks. And what we wanted to look at were skate embryos from nursery areas in the Bering Sea. And we had several research questions, and there's just a lot that we don't know about skates. We wanted to know are the nursery sites genetically distinct, so that they should maybe be protected? And that was the main research question. And we looked at, I'm just going to talk about Alaska skate. We actually looked at another species as well, but just for the sake of time, I'll cover Alaska skate. So this is what a skate egg case looks like. I'm sure you've all seen them. But what's really interesting and surprised me is that they gestate for up to 5 years. There are distinctive spawning sites, or not spawning sites, nursery sites in the Bering Sea where you'll find maybe just one species egg cases or several. You can go back year after year and find the same species egg case laid there. So we took samples of Alaska skate egg cases with embryos from a series of known nursery sites along the Eastern Bering Sea shelf. Again, here's Pervenets. This was just called IPZ 1 and 2, Pervenets Canyon and Bering Canyon. And we did rad sequencing. The skate genome is a lot bigger than fish genomes, like by two or three times. So I allowed for that. I got more loci. Oops. Well, it depends on how you filter it, but I got quite a few loci. And I sequenced 479 skate embryos throughout the range, also 44 adults, and then another species called the leopard skate, which we'd recently discovered in the Western Aleutian Islands, kind of just for comparison. So when I first did that DAPC plot, remember I showed you that before with Pacific cod, and you have individuals that kind of separate out based on how different they are. The sample really stood out. It was from Pervenets in 2016. But what was weird is we had a Pervenet sample from 2007 that grouped with all the other Alaska skates. And I was just, what is going on with that? So I did a lot of kind of thinking, and I came up with this plot here. And this comes from comparing this estimated kinship coefficient that is from a program called King, that if you plot it against the estimated probability, that this is kind of an esoteric genetic term, that identity by state is 0. You don't really have to worry about it. But what you get are related individuals grouping together. So it's kind of a sort of common method of looking at relatedness. And in this case, I'm going to show you the green here is Alaska skate adults from the Eastern Bering Sea. There's gray behind it that are also Alaska skate. And you'll see where I got these samples from. Green are all Alaska skate. Remember I said I sequenced a leopard skate? So the red here is the Western Aleutian leopard skate. And here's where the leopard skate pulls out. It's definitely a distinct line from the Alaska skate. And then here's all those Pervenets 2016 individuals. The fact that the Alaska skate and the leopard skate are known to be different species made me think that we'd kind of happened across a new species here. We don't know much more than that. And it's hard to tell a lot from the embryos and the egg cases. So I think there's more work to be done here, certainly identifying what the species is, and maybe if we can find an adult of it. But this is kind of one of the really cool things that I get to do in my job, is sometimes find maybe a new species. Oh, another really cool thing that came of this was I did look at genetic distinctiveness of the different nursery sites. I can talk to that, but I really highlighted some different parts of the study that I didn't really know were going to come out of it. So remember I said that they sit in their egg cases and develop for 5 years. So you can take a look at the embryos, and 1-year-olds are like this big, and then you can kind of tell how old they are. So we could stage them. So I'm going to tell you, like, a stage 2 is a 2-year-old, stage 4 is a 4-year-old, stage 5 is a 5-year-old. And then I could look at relatedness among them. And I found a surprising amount of relatedness. I didn't find any full siblings though, but I found half siblings, which are represented by green arrows among individuals. And the individuals just have these boring 6,000 numbers. So the individuals with green arrows among them are half siblings. and the individuals with orange numbers between them are cousins. And so in this one nursery site, this is actually the same sample from Pervenets Canyon in 2016, you could see there was half siblings and cousins, which meant that the adult, and I can't tell you if it was the male or the female, but the same adult returned to that spawning area multiple times. This is where the site fidelity and the faithful comes from. But I think that's really cool because it lends to our understanding of the reproductive system of skates, whether they have site fidelity. Yes, I'm showing you that they do. And also evidence for potential genetic distinctiveness. And I didn't make a slide of it, but I did find genetic distinctiveness among some of the nursery sites and not among others. Some of the ones Bering and I'll show you up here. I didn't find genetic differentiation among Pribilof and Bering Canyon. And Jerry Hoff, who's kind of the expert on skates, thinks that they just kind of spawn lay their eggs all along here. There's not really two distinct nursery areas, but the others did appear to be distinctive. So that's all I have. But I hope that I've shown you some ways that rad sequencing and next generation sequencing can be useful to answer interesting questions and get at maybe biological questions that you couldn't get at other ways, answer questions about pertaining to fisheries management. I'm excited to see where you all take this with halibut. So with that, I'd be happy to answer any questions.
Thanks a lot, Ingrid. That was great. So the floor is open for questions.
Hi, great talk. That was really wonderful. I have a question. So with the first study you were talking about with the cod spawning, you're talking about using the same SNP panel between different species when you were looking at their, so when you're using the different species for this, so you're saying the same restriction enzyme and the same restriction sites for those SNP panels, how different are those species? When you say species, how closely related are they when you're looking at those
so
SNP panels?
That was something I might have mentioned in the introduction, but it didn't have anything to do with the Pacific cod work. That was all Pacific cod.
Correct, yeah.
But I have done another rad sequencing study on snailfish where we actually used rad sequencing to do phylogenetics.
Okay.
And we had like 20 species, I think. And we were able to compare across species and find the same sequences in regions that I think were conserved between the species. But what happened is that you ended up getting fewer and fewer sites because I think we just only ended up with like 300 in the end.
Oh, wow. Okay.
Yeah. So because there just weren't that many sites that were found among all those different species.
Okay. And that was kind of my question regarding, because you're saying that was plugged into the model with it looked like, you're plugging that into the model with those different species. And that was kind of my question, is how much overlap can you work with when it comes to those SNP panels? Because my thought would be that there wouldn't be much overlap. Which is
and the more distinct the species are, the fewer levels of overlap you'll get.
Yeah. Okay. Yeah, no, that was very interesting. So at what, I could go on forever, I guess, but I guess with 300 SNPs, what kind of, I mean, that's, normally you're looking at SNP panels that have thousands or tens of thousands. What level of specificity of questions can you really look at when you're using only like 300 SNPs?
Well, in that case, we were interested in understanding phylogenetics of a bunch of snailfish species. And so we compared a phylogenetic tree based on that data. And in that case, we actually used a whole sequence, not just individual SNPs.
A marker,
and compared it to other trees that were built with, one was with cytochrome oxidase. And what was really interesting is the trees were almost congruent. And so they kind of lended support to each other. So even with 300 sequences, it was just as good. I don't know, you can't say if it's just as good or better or worse, but it's quite similar to with another kind of well-documented method for building trees.
No, good old Cox won. So
Good old,
Yeah.
yeah. Thanks. Alan, hi.
Thanks, Ingrid. It's I just love your excitement about this field all the time. Every time I see you talk, and it really brings me in. So thanks for coming out here today.
It's so cool.
It is. It really is. I mean, I'm so far behind too. But I'm wondering, and really not related to your research, but when you were doing this research, have you ever found, or have you ever heard of anything that indicated that that these range expansions of cod have occurred in the past? For example, have we seen cod in the Northern Bering Sea in high abundance?
Really good question. We've looked at all the studies or all the surveys that have ever been done in the Northern Bering Sea, just to kind of go through, like, were there really never any cod up there before? And the answer is no. Handfuls of baby little ones here and there, but nothing more than that. And I really probably need a slide on that because you're not the first person that's asked me that. But Dwayne Stevenson helped me go through all the old surveys and sort through, well, is this really the first time? And I think really, yes, it is the first time in modern history anyway. Yeah. And really the predominant, there's Arctic cod was the predominant species there. There really weren't many Arctic cod found there in our last survey. So things are really, really shifting. And yeah, it's not just this species. One more, yeah.
Thanks again. And with that being said, were you able to find, or did you actually look for any evidence that maybe the Gulf of Alaska cod were replacing we're coming into the Eastern Bering Sea, and the Eastern Bering Sea were moving to the Northern Bering Sea? Was there a wider range shift than just Eastern Bering Sea to Northern? Maybe the Gulf cod were coming up through there.
That is such a cool question. Yes, so if I had really old samples, then I wouldn't know. But luckily, the Unimac Pass sample that I used in the study was from 2018. And it was, you saw it, it's distinctive. Didn't look like what the Kodiak signature was. But that's a good question. And I actually was pretty active getting samples this year. So I have samples from Kodiak, Shelikof, Prince William Sound, and Unimac this year. So we can compare over years to see if the kind of the baselines have changed at all. Yeah, and I think there is some connection between, fish, maybe Kodiak Island westward. That's something we're still kind of investigating. Like, we know that Shumagon Island cod seem to move into the Bering Sea in the summer, even though they're managed as part of the Gulf management unit. Yeah. Yeah. Yeah.
Any more questions from the audience here? Do we have any questions from webinar attendees? No? I have a couple of questions on the pollock temperature experiment. And that is, first, was that done with whole genome sequencing, or
rad
was
sequencing.
that rad sequencing?
Yeah.
Rad sequencing? Okay. So you're looking at the presence of loci in the fish that survived the temperature treatments, right?
All three treatments comparing, yeah.
Right. Yeah, the question is whether, for instance, in those samples, you had the chance to look at the expression levels of all those genes, because the change in allele frequency may not really be functionally relevant.
Exactly.
Right?
Yes, and so this got at just, what is the genome? It's not giving you anything about transcriptomics, but I think that would be a really important next question, is as to why. What is the benefit?
Right, because maybe those particular genes are actually maybe in a part of a linkage group or set of chromosomal regions that are really selected or indicative of temperature adaptations, right?
I'm not sure it was the best designed experiment, but I was thrilled to find something out of it in the end.
Experiment, yeah. Any more questions? Nobody? Well, thanks very much.
Thank you all.
That was really interesting. Thanks.